it's already started.
Okay, very good. Well, good afternoon. I'm Rick Hamilton. One of the great things about being new into a given field is you get to ask a lot of dumb questions. And I know that many of the challenges facing FUS today are not technical. Many of them are related to inertia, related to reimbursement, related to a lot of other factors. But I had to ask the question, what are the technology gaps that need to be filled in order to accelerate the adoption of FUS? And fortunately for us, we have brought together a very esteemed panel who can help answer these questions. And so I plan to take a lot of notes from this session. So if you see me scribbling furiously, you know why. So with that, I get to introduce the symposium president. I'd like to turn it over to Alyssa Katafagu and the Technology Gaps panel. The floor is yours.
Thank you so much. I know some people are still outside chatting, but we have to get started. And we have great panelists both in person and, of course, virtually. We're very lucky to have people from industry and academia with us today to see where we can find the gaps and then hopefully find some solutions for some of the gaps in technology. The previous session was very informative. There were some aspects that were already touched upon, but we can expand some more and maybe talk about different things that we feel that we have to dedicate the next 2 or 5 years to be able to have this technology translate better to the clinic or make an impact with pharmaceutical compounds or other applications that we're thinking of. I want to echo what the panel mentioned in the previous session, that this is an absolutely exciting time and also a very kind of disorienting time about where we can go with focused ultrasound. It used to be a few years back that all of us would show some ablation slides and that we can lyse the tissue and show some apoptosis, and now we have veered off more into the molecular world, some of the genetics world, and looking at what expressed what immunological applications we can think of. So I think it's an explosion, but at the same time, we need to see and prioritize potentially where we can make the highest impact. So I think that's basically some of what the panel is going to discuss. And I would definitely want the audience to be engaged. If you have a burning question, or even if you have a suggestion for the panel, or you have an idea that you want to have a steer towards and get the panel's opinion, I think this is the time to do it, and this is the right forum. So definitely online and also from this room, don't be afraid to ask any question. So with that, I would like to start with asking all of the panelists, starting with the in-person, to introduce themselves within 1 minute, their expertise in focused ultrasound, and maybe also what gap they think we can start with just one gap for now. Thanks. Okay, you can start. Andreas.
So it's me, yeah.
Andreas, yeah.
So I'm Andreas Melzer. I'm founder of the Institute for Medical Science and Technology at University Dundee, and we made our first attempt to focus ultrasound in 2009, and one of the key projects we started with was drug delivery, was called nanoporation. Got a success story as an EU project, and one of the key developments, which I think is still a gap, was cell culture sonicating, and not just a cell culture. The cell culture, people have 96 well plates for high throughput screening, and this is something we tried to establish. We were not super successful, but more recently it went better. Another gap was moving organs under MRI guidance, a very significant problem. We had a very large EU grant with Insightec and other companies involved, and we made it somehow working, but it's still far away that we can under MRI guidance, sonicate the liver through the rib cage without harming it. So this is one gap, I think. And another gap, when I moved to Germany, I still have some function in Dundee, but my main function is director of the Institute for Computer-Assisted Surgery at University Leipzig. and we went into the combination of radiation therapy and focused ultrasound, which I think is a completely underdeveloped area, but not us found very interesting results, but others as well. And last but not least, the monitoring of focused ultrasound effects, although we say MRI, but what you see is temperature rise, not extremely reliable, and then an edema kicks in and some stop of perfusion. But have we killed tumor cells? The only way to see this currently non-invasive is PET imaging. So we're setting up in Leipzig a PET MRI-guided focused ultrasound setting, and at least we have the technology ready, but we haven't done any clinical work. So I think I'm over my time. And to Peji.
Hi, everybody. Thanks for including me on the panel. I'm Peji Ghanouni. I'm a radiologist at Stanford. In terms of our assignment for this, when we were preparing, the question was posed of if there were no resources, no limitations on resources, what would you want? And that's hard to summarize in 1 minute. But I can tell you,
we're going to go back to that. This is just for your introduction.
Yeah, but for the introduction, I think the point I'm trying to make is that the limitation that I see is a very broad one. It's that we are too constrained by the forms and software and planning behind the devices that we have. I exclude too many patients from the treatments that we would like to be able to offer because of these technology constraints. I've tried to treat sarcomas, the pancreas, gynecologic and urologic cancers. We are asked to treat spinal mets. We can't do many of these things safely and effectively because of our device. And I would say that as a result, we see conversations about ablation plus something. Let's add some other adjuvant therapy on top of the ablation because the ablation doesn't work well enough. But if it did work well enough, we wouldn't do that. If the chemotherapy worked well enough, you wouldn't add radiation on top of it. But it's because the primary treatment isn't working well enough that we're looking for these other options. And I think that the point should be, how do we get that primary approach to work well enough where we don't need anything else?
Hi, I'm Yoav Levy. I'm the Chief Scientific Officer of InsightTech. Thank you for inviting me to this panel. So I'm in charge on all the non-clinical research at InsightTech, which means acoustics labs, MRI sequences, and all the animal work that we do. I'm kind of working in stealth mode, not intentionally, but practically. So I used to work with Andreas and also with Peji, but it's first time we really meet. And also visited a few times at Sunnybrook when trying new features that we developed.
Okay, great, thank you. So then we're going to go to our virtual panelists. We can start with Gary Choi.
Hi, everyone. I wish I could be there in person. I'm a diagnostic radiologist, and I still practice at the Palo Alto VA, and right now I am at United Health Group in corporate medical affairs, all focused on clinical transformation. So our group looks at how we can do things better for our patients and physicians. You know, United is not only an insurance company, but we also have Optum, which is a care delivery organization. And we also have services. So really across United Health Group, how can we start to incorporate technologies that are value-driven, that are practical, that are high quality, effective? And as a little bit about myself, I used to be an engineer, chemical engineering, and actually spent some time at the NIH back in the days. I guess I'm dating myself, but 20 years ago, 2002, I was first exposed to high-intensity focused ultrasound, working with Brad Wood and some of his colleagues at the NIH in mouse models. And seen it come a long way in practice. You know, RFA and other cryoablation techniques. I trained with Peter Mueller at Mass General, doing a lot of these procedures myself. And now I'm in a role that's in industry, so to speak, and would love to see the continued advance and incorporation of, we've made progress, incorporation of focused ultrasound in practice and within, you know, more clinical applications in a practical, you know, affordable way. So super excited, and it's great to see familiar faces here, including Brad.
I guess then we're going to go to Brad because it's a perfect segue.
Thanks, Gary. Good to see you too. So I would be remiss if I didn't start by tipping my hat to my first mentor and the reason I went into medicine, Neil Cassell, who's such a visionary in terms of where he sees things going and where he saw things going so many moons back. So I think I owe him a huge debt of gratitude. So I run and set up and run I take credit for the team science of a public-private, multidisciplinary academic industry partnership that has a lot of different disciplines represented at the NIH Intramural Research Program in the Clinical Center and NCI, and a thing called the Center for Interventional Oncology. And essentially, I'm blessed to go to work and learn something new every day from different disciplines. And I think over the years, we've kind of gotten some interesting perspectives on translational science and what it takes and helping to define the limits and the gaps and the places and pitfalls that people and technologies and approaches fall apart. So I'll leave it at that. But I think we've been playing with HIFU in and out. It's one of a number of different things we do. I'm a clinician at heart. And we was lucky enough to do some of the first ablations in patients in certain organs and settings, which is neither here nor there. But I think the translational piece is what I'm sort of most interested in and most energized by, and look forward to this session, really.
Okay, great. Thank you, Brad. And I guess I'm going to go counterclockwise on the screen. Graham?
Hi, good afternoon. It's Graham Woodworth from the Department of Neurosurgery at the University of Maryland. I'm a brain tumor-focused neurosurgeon, and I started working in focused ultrasound in 2009, preclinically, trying to find ways to get therapeutics deeper into the brain, to regions where we couldn't remove the tumor cells that were really woven into the fabric of the functional brain. And it was that insight and the opportunity to potentially disrupt the blood-brain barrier and deliver therapeutics into those regions that really weren't accessible safely with surgery that really inspired me to move into the translational and clinical realm with the technology, and have been involved in a number of clinical trials now, leveraging focused ultrasound mainly in the blood-brain barrier disruption mode with microbubbles. And we've continued to do that today and expanding those efforts here at the University of Maryland and with our colleagues across the country, University of Toronto, Sunnybrook, and Brigham and Women's Hospital and elsewhere. So in terms of a gap that I see, and hopefully we get to discuss, is really increasing in vivo readouts of the focused ultrasound bioeffects, rather than just using new contrast enhancement on MRI to evaluate, for example, some form of efficacy, and maybe T2 star weighted imaging to evaluate some form of toxicity if we think that's what it is. It'd be nice to have other readouts of bioeffects, whether those bioeffects are desired amounts of drug delivery or given a focused ultrasound effect that we are having, either acoustic effect or mechanical effect or a thermal effect. So if we had that, I think we'd be able to go a lot deeper into the field and understand more about what we're doing mechanobiologically or with the focused ultrasound from a therapeutic perspective.
Great, thank you, Graham. And last but not least, Kullervo.
Hi, I'm Kullervo Hinnanen from Sunnybrook. I'm a physicist, and I started with focused ultrasound with my PhD studies 1980, and I haven't finished my thesis, so that's been long road, maybe too long, from basic physics, simulations, transducer equipment development, animal testing to clinical trials and commercialization. So kind of the full spectrum there. And what is the gap right now? I think we need to get focused ultrasound adopted to the clinical use so it becomes treatment of choice. And one thing is, of course, is the treatment times are still quite long for ablations, so getting them faster would make it better. And then if you could get diagnostic ultrasound guided systems, that would be good. But that would require that we can do 3D imaging of the ultrasound field throughout the whole beam path where we are using ultrasound, where the therapy beam is propagating. So that's quite challenging technically, but maybe it can be done.
Great, thank you so much for this great introduction, especially setting the gaps And I'm taking some notes. We're going to go back to them, or feel free to bring them up. So we have 4 questions that we came up with, and it's we have paired them for who is going to start, and then we can have a discussion. So the first one is, if we can identify the current clinical advantages and limitations for both MR-guided and ultrasound-guided treatments, and how we can improve those systems for faster adoption. So I think Graham volunteered for that, and we volunteered also Kullervo. So maybe, Graham, you want to start?
Sure, thanks. So many of us are using the Insightec system, but there are a number of other clinical systems that we've heard about in this conference today, especially the Navi FUS system, the Carthera system, and some of the others that are coming into practice now. So I think that we're seeing that these platforms have different strengths and advantages, and I think part of the issue that we need to solve as a group is what biological impact or effect are we going for, and how can we, as I mentioned before, get a readout of that effect. And I would say, from my perspective, new contrast enhancement on the MRI is not enough. We need to know more about what we're doing so that we can compare the blood-brain barrier opening that would happen with one of these tools compared to another in a more accurate, effective way. Whether that readout is a desired amount of new drug delivery of a given therapeutic, or a desired amount of immunomodulation, whatever that is, we need to know how to compare the end effect better across these different tools. So while I think it'd be very, very nice to speed this up and be able to do this faster for patients, we've treated many patients with this device, and yes, it's taken hours in some cases to do that. I would be okay with that because I want to make sure we know what we're doing. And I'm not convinced we do. So I really think we need to get some of these better readouts of the actual biological effect that we're having in and around the body, and especially the brain, because we're talking about working in highly functional areas. And so I think if we can design our tools and our downstream assays of reading out the bioeffects that we're creating, I think that'll be very important to the development of the new technologies. I think in the next few years, we're going to see a rush of new companies coming into this space. And I would say we got to be very careful on the so-called Gartner curve, which is where we are now, on the rising up on the peak of inflated expectations per se. And we need to make sure that we don't overdo it and hurt people. And so I think whether it's blood-brain barrier disruption, thermoablative uses, or in between, or histotree, I really think we need to be able to get to a point where we have comparable downstream assays or tools to assess the effects we're having, so that we can compare even the tools we have now. How do we compare what the Carthera blood-brain barrier disruption is to the Insightec blood-brain barrier disruption, other than some amount of new contrast enhancement or dynamic contrast imaging or something like that? I mean, I would say that's not enough. So I would say those are the biggest gaps and issues that I have with where we are today, and what I'd be a little concerned about as we bring new tools into the equation or into the arena and start using them in similar ways, thinking they're having a similar effect just because we're seeing contrast enhancement. So that would be where I think we are with our clinical gaps and tools right now.
So to understand, Graham, you think that with the MR-guided, the pivotal moment is to get a better readout for faster adoption? Is that what you think is needed?
Or understanding better what we're doing, because we're using the acoustic emissions data, in the microbubble mode, and the new changes on the MRI to assess the effect that we've had. But I think we need a deeper understanding of that effect and a way to compare it head to head, whether it's with those two devices, of the acoustic emissions detectors and the MRI. That's fine. But again, that gets us into the MRI world, and time, as Kullervo said, is of the essence as well. So can we get, if we could get outside of the MRI world to assess these effects and then be able to compare them across the different devices, that's where I think we need to be going, so that we can, again, get to the point of making them faster, more efficient, and more reproducible in the patients.
Okay, great. And Kullervo, you mentioned the treatment times that you think is the biggest gap. So that's probably something that we can start with and see whether MR-guided can actually become faster. Do we have to switch to ultrasound-guided for some treatments? Or where do you see we can get faster treatment? Where is the main bottleneck?
So maybe to answer kind of what Graham was saying, I agree with having the readouts. But I think that's mainly in the brain where we need better things. I think the ablation side, like Elisa has done stiffness mapping and things like that. I think there we can have good enough readouts with MRI or ultrasound to be able to tell what has been ablated. So going back to the going faster. So with blood-brain barrier opening, I think that would be a solved issue in few years. With the phased arrays, you can go really fast, electronic beam steering, so that will not be a problem. Can you make the permeability enhancement uniform? I think so. In animals, we definitely can if you do a precise point control. But there you need to really image where the bubbles are and how they are performing. And in animals, we can image them with a million volumes a second throughout the whole thing. If that can be done in real time, then we could control it really precisely. And I think that is the technical challenge. There's a huge amount of data that you have to collect, and we need to be able to solving about all vibrations and super resonant population locations. So that will take potentially artificial intelligence to be able to do fast enough. So that is definitely a gap there. With the ablation outside the brain, we need different devices to do it faster. Just going point and shoot will not get us fast enough for MRI. If you're using ultrasound guidance, then maybe it's okay to have 2 hour, 3 hour treatments. But the disadvantage of ultrasound guidance right now is that we cannot see the ultrasound propagation path like we see with the MRI. So you can identify if you have bowel loops or anything in the way. So that's a big advance with MR. I think it can be solved with ultrasound also, but requires something else than using just commercial probe that is planted in the middle of the array. So that's kind of my assessment right now. So, thanks.
Thank you. So advancement on the hardware. Yes, sorry. Yes, please, Brad.
I was just going to say Clairvaux and Graham really epitomized the representations of a clinician in the biology and the physicist, acoustic physics in the underlying mechanisms and the physical sciences. And getting those two together is so key and also critical to understanding the benefits and potential advantages specific to HIFU. So the parameter space is so huge. I mean, Clairvaux is single-handedly responsible for him and his teams of unearthing and addressing a lot of the mechanisms, underlying effects given certain parameters, whether it's mechanical, acoustics, or heat or whatever. And this give and play between clinician and physicist that we just experienced there, I just didn't want to pass without commenting on it. That's really critical to everyone's understanding of this whole thing. And therapeutic ultrasound itself, the benefit is the fact it's also its weakness, in that we don't understand the multifactorial, multi-parameter space and their bioeffects that well yet. We're starting to, but that's also the beauty of it, because you can tweak and potentially tune it in a way that might have an intended bioeffect to meet a specific clinical need in a specific unmet situation, which we've been trying to deal with as clinicians for so long. So I just didn't want to let that point pass. I'm sorry to interrupt you.
No, not at all. I mean, that's the nature. The discussion is to be actually dynamic and spontaneous. So I think we can.
Let me interrupt you some more then.
Yeah, good. Please do. All right, so we're going to move to the next one. I think that was very well said and very nicely summarized. Thank you. So the next one is more philosophical, and I think Peji actually already mentioned that, which is if we had unlimited resources, and it is a big if, what would be our priorities for basically the ideal focused ultrasound system, and what would it look like? So with that question, I go to Yoav, and then Peji after that. And feel free to jump in after, for discussion.
So I will refer also to the things that they discussed right now. And I'll start with the commercial systems that we have that treats the essential tremor. And one of the limitations that were mentioned right now are the treatment rate. And in another aspect, we were talking with Peggy, and he was concerned also in public from the spot shape. And I think that this is one thing that still can be improved. The spot shape can be improved in our commercial system, and that might shorten the treatments once the physician have more confidence in the spot shape. One important building block for that is volumetric thermometry. We right now provide multiple slices in our system, but internally we already developed accelerated volumetric thermometries that can give 5 slices of 2 millimeter in 3.5 seconds, but the limitation in putting that in the system is just to convince the MRI vendors to put that in the MRI that you use together with our system. So assuming no resources problem, I think we could convince them to do that. Additional feature which is probably not needed usually, but we also developed the option to do eco-focusing, which is focusing based on measurement instead of focusing based off a prediction. And we actually took it to a clinical trial. And what we find out is, first of all, that our city-based focusing is pretty good. So there is no use case in order to put it on all our systems, but it still can be helpful in challenging cases, and that can also improve the spot shape. So that was regarding the treatment rate in the current system. Now, moving to the low-frequency system, which can be considered almost product for drug delivery. I called it almost product because I'm sure that it will work at the end. But, and it's a big question, not question, but challenge, that we should measure our product according to the clinical outcome. And we are not there yet. Regarding the efficacy of the clinical treatment in all those drug delivery applications, there are still many unknown variables. Which drugs are going to benefit from such a system of such a capability to cross the BBB and give a good outcome to the patient? How much margin should we take to cover? I was surprised that I never got a request to support treatment of a tumor of, let's say, 3-centimeter tumor and the 3-centimeter margin. Altogether, it's 350 cc. No one asked me to support such a treatment so far. So physicians don't go there yet. How aggressive should the opening be? How many sessions does the patient should go through? How critical is the homogeneity of the opening? Can we open just 50% and that will be okay? So there are so many known unknowns, and on the other hand, each experiment is so expensive. So our strategy is to get to the experiments, to the clinical trials, with systems that have extra capabilities, Therefore, we maximize our chance to succeed in the treatment. What we try to bring is systems that have faster treatment rate in order to manage to treat much bigger volumes. Let's say with the know-how that we have today, we can at least multiply by 4 the treatment rate. Also, while preserving our unique ability to give a homogeneous dose coverage. Also, we would like, and doing so, but improving in each version, to improve our intraoperative MRI imaging, especially the T2 star, that can, along the treatment, validate, that confirm that we are treating in at least reasonable aggressiveness. Therefore, the patient would be able to focus on the efficacy of the treatment instead of being concerned from the safety of the treatment. Once we find the application and the condition in which the patient get benefit, then we can go to optimize the treatment flow, the cost of the system, and all of that stuff. And I can assure you that getting out of the MRI will be much easier than getting into the MRI.
Thank you, Yoav. On that note, we're going to turn to Paige, and then maybe you can give us the clinical side of things, of the ideal system.
So I don't know how many of you guys are Star Trek fans. Show of hands. Yeah. So somebody goes to McCoy, he puts him on the bed, and there's this semi-cylindrical thing that folds over the patient. It figures out what's wrong with him, and it treats him.
Yeah.
Right?
There you go.
So imagine that this is something that allows coupling to a device that is filled with whatever density of ultrasound elements that Clairvo can possibly pack in there. So, and it's coupled to some imaging tool that allows us to identify, maybe based on preoperative imaging as well, where the lesion of interest is. And Clairvo's got so many elements and whatever supercomputer he needs, so he can get this ultrasound, a conformal treatment around the lesion without worrying about the intervening bone or the bladder that's next to it or the rectum that's next to it or the nerves that are next to it. And it's one position for everybody. I don't have to contort my patient to fit his hand or his ankle or the pelvis or the breast or whatever on top of the device. It's a one-size-fits-all. Everybody gets coupled to the thing. I can see the lesion, and then he can deliver a conformal treatment based on where I tell him the lesion is to the spot. And I don't have to care about what's in the way, about scars and bowel and so forth. And maybe on top of that, I don't have to monitor the treatment. We don't monitor gamma knife treatments. We don't monitor radiation treatments, right? We deliver the energy, and we know that that dose is sufficient to cause the results that we want. And the patient's gone, and later on we do a PET scan to see whether the tumor is dead or not. We worry a lot about this, the monitoring because of safety. But if we knew more about what treatment parameters were necessary to achieve the ablation that we need, then we could deliver that energy in a conformal pattern without worrying about safety around the area. Obviously, we're not there yet, but you were saying I'm supposed to dream, so I'm playing the role of the crazy dreamer on the panel. But that would solve a lot of my problems.
Yeah, I agree with you. Star Trek is very inspiring, and we should all go and get our ideas from there. But before we do that, any other ideas from the panel, how we can change things and go forward?
I think you got to be careful. I love McCoy and his telecorder or whatever it was.
Yeah.
But we have to be careful. It represents a bias that we all try to stuff things into a model, and it has to be perfect. It has to all be portable. It has to be something that solves all problems or meets all clinical needs or solves every little thing that we view this magical therapy to be. And just have to be careful because you have to fight against that desire to have things be perfect, because in some ways, because to be devil's advocate, because all you really need is to have a single linear line to benefit and value that makes commercial sense and is cost effective, has a business model that works, reimbursement plan. Otherwise, you can spend decades and decades trying to get perfect. And all you really need is that one little, and it's starting to come. We've listed some of the FDA approvals at this meeting, and these are starting to chip in and we're starting to chip away, but just a word of caution against trying to make things perfect as we're trying to get to the ideal world.
Yeah, maybe I'll chime in there, Brad. I totally agree. I think that applies to a lot of technologies. Like we will get to perfect, but where do we start in an incremental way? And there are multiple beachheads you can keep advancing once you hit a practical beachhead where there's efficacy, safety, reimbursability, and then prove a lot of benefit there, and then move on to the next target. It's a target-rich environment. Which is the right target to point focused ultrasound at? And I think over the last decades, right, given a lot of the great clinical and scientific advances, we basically are now seeing focused ultrasound being used more and more for essential tremors, bone mets, et cetera, continuing to win those beachheads, to hit those targets, I think will be good for the field. And of course, the technology should continue to improve there so that more clinicians could be delivering this at the bedside or in the OR. And then continuing like the focused ultrasound under Neil's work to really bring people together has been very impressive to see. And that work has to continue to happen. More research, more community to get to the Star Trek vision. Because Star Trek is pretty cool, and I think that there's a lot of reality there too. And I think incrementally we can get there. But there are a lot of targets that we can hit in that journey.
Yeah, it was a philosophical question, but since I have both Brad and Gary unmuted, what can we learn from other fields, surrounding fields, for faster implementation? What can focus out? I mean, Paige mentioned radiation. So what can we learn from those fields in order to get to the next level with focused ultrasound? Maybe Brad. Yeah, Brad. Yeah.
I think taking a step back and realizing that not all scientific questions coalesce and align with the actual translation. And so that you have this intellectual curiosity fighting with practicality of trying to make a difference and have impact. And they're always fighting with each other. And we can learn from other disciplines. There's a million ways to approach oligometastatic disease or locally confined tumors in any organ, a lot of different organs. And there's a whole bunch of different technologies out there, and drugs, devices, image guidance, radiation therapy, microwave, cryo, RF, pulsed electrical fields, IRE, laser. There's just, PDT. There's a lot of choices out there, and each of them have their advocates. So figuring out exactly how we can have this coalescence come together is going to require learning from everybody else's failures along the way, but also learning about how they can move fast. So you have, urologists in the prostate that move really quickly towards certain technologies. So if you can give them enough of a bone to have them all fly that direction, win. And I think there's all kinds of different examples of that learning from other disciplines, learning from other energy applications is going to help us coalesce this giant effort. We've talked about the physics and biology coalescing. Well, the same is true of clinical sciences and a lot of different disciplines coalescing with the business plans and the commercial factors. So, in short, there's a lot that we can learn and more than we can talk about in this session from the many different disciplines, whether it's microwave versus RF in the liver, whether it's cryo versus microwave in the kidney, whether it's, every different organ has its different application and its different subspecialties that creep in, and they all have different phases of development. So it's not just, we're not in this alone, and focused ultrasound can't look just within its own community to solve it.
Great point, Brad. I would add, we can look at close to radiology or imaging, like let's say diagnostics on the non-interventional side. I think we can look at the successes of CT, MR, even diagnostic ultrasound. It's become a tool you can't live without. It's like you can't live without it. Like, could focused ultrasound one day get to that point? Can't live without. Plus, there's trust, right? Whether it's safety, I think Graham mentioned consistency in results. You just know when you put somebody in MR or CT, you're going to get images that look pretty good. Philips Siemens, GE, pick your favorite vendor. It's kind of become commodity, right? Like you expect an X-ray to look a certain way. So could focused ultrasound become the can't live without for XYZ application, that you just trust it. You know, you trust that it can do its job, and you trust that it's safe. And you trust, and it can be operator independent as well. You don't need a superstar interventional radiologist or neurosurgeon to do it. Any, like a trainee will end up trying to do it. You know, you trust that somebody could use it.
So if I
That
can,
standardization point is key, and we undervalue it. If you can have a closed loop, and we can develop more closed loop mechanisms to make this one therapy that anybody can do, then you have an advantage over a lot of the others. And standardization is going to be more important in the future.
So standardization for trust, that's basically how I can summarize this, that we need more standards on this. Okay, so we're going to move to the last question, and then we can, if we have time, we can take some questions from the online, or people can go to the microphone. So what technology should be developed for efficient / real time, and that could be for all the phases, from planning to targeting. Okay, maybe monitoring. I know Paige says it's not needed. An assessment of the treatment or whatever effects we intended. So Andreas, you want to talk about that?
I think it's an important question and relates to several of the things already said. And to be more efficient is probably something to reduce all the monitoring effects we undergo. And although the comparison to radiation therapy is a bit unfair, when you look to the history of radiation therapy, the patient had to pay a huge price for this trial and error of radiation therapy, but they learned the trade, and now they apply a dose. And ultrasound thought about, we need also a thermal dose to apply. I'm not quite sure whether this is what's really needed. We need to identify better the outcome of the treatment, and that's why I'm trying to get in a PET MRI a setup, which likely will never be the routine. However, if we can use PET in conjunction with MRI to really understand how long do we have to sonicate and at which energy levels, pressure levels whatsoever we sonicate, and then we can move more to these more efficient radiation therapy setting where we have a certain area, a certain tumor, and we know what's going to happen, and we have proven the results a bit more efficiently. When it comes to targeting, we had in the panel before, the other panel in the morning, the question I raised about ARFI, acoustic radiation force imaging, which has been around for a long time. So how and when can it be implemented? I think it's really an important tool, but currently energy levels you have to apply for getting a good ARFI MR imaging is too high. So we should team up better with the imaging companies, with the MRI imaging companies. And as you may know, Siemens Healthineers has really put this on top priority, and they do quite a lot of work to help clinicians. So we should get them on board and say we need ARFI at a more sensitive level so that we can use ARFI for targeting. And it will also improve temperature mapping. There was mentioning temperature mapping is about to be improved, and there are also things around for a long time, and this needs to be pushed further so that we get a more precise understanding what exactly happens at the very spot. And the same is for monitoring, assessing, targeting, assessing the treatment. This is what I mentioned before, that we have a more comprehensive assessment what really had happened after the sonication, what kind of effect we have achieved. And this is where, again, PET kicks in. And Paige mentioned we do our cancer patients, they get after a certain time a PET. But I'm not aware that many of the cancer treated with ultrasound are integrated in the schedule. So I can only motivate anyone who has access to a PET MRI to go that route and try to convince the involved people and nuclear medicine people to develop better monitoring assessment techniques for the targets, and particularly tumors treated. Yeah, that would be my...
Great, thank you. I also agree that, monitoring is needed. And plus, we've learned from ultrasound itself. I mean, imaging, for example, where I originally came from, that you can do real time, and I think that's the advantage. I don't know how radiation. I don't think we can do ultrasound imaging on radiated material. So the technology doesn't exist. But since we have it, and there's a lot of different effects that can be instilled in the brain, such as the radiation force, temperature. The blood flow changes are very interesting, and fMRI, for example, could be potentially used, but there's also functional ultrasound and cavitation, of course. That I think this conference has been very informative about how we can actually take cavitation to the next level, and mapping has been very promising by multiple groups. So I think we have an advantage there to potentially do this all of the above in real time. In the brain, it's tough to do everything because of the skull, but you can do it in other areas, and that could be linked with some of the outcomes. So I think it's an advantage, the fact that you can have those in tandem together, working confocally. We have a couple minutes left, and everybody's very shy in this room. So I will go back, I guess, to Yoav and ask more about the focusing abilities, because what is the perfect focus? Is it going to be the 300 cc or the 1 millimeter cubed? What's the perfect focus you think that is needed? What are you going for?
When we talk about focus...
treatment volume. I'm sorry, treatment volume. And focus that comes with the fact that you can do it potentially in one shot.
Okay. I think that we should... the two things are related, but they are not the same. We should treat big volume, but we should use high resolution to treat that volume, because in that volume you can have many types of tissue, and even within the same tissue, there are areas with one vascularity and areas with other vascularity. So optimal solution would be something with very high resolution, let's say millimeter on 3 millimeter, and something that can scan with raster or no raster a bigger volume in order to cover whatever needed, whatever requested by the physicians. So we need something with a small f number to get high resolution, and also something with electrical steering that can work very fast and cover big volume with real-time analysis of whatever feedback that we get from the tissue.
So I circle this back to the clinical panelists. Anybody want to take the clinical version of this? Question?
I think I'd be remiss not to mention immunomodulation as the tip of the iceberg, and what could really change the face of oncology if it's worked out appropriately and in the right fashion. So, I think we just have to get folks on the same page with defining a pathway, and again, learning volume. We chased volumes in liver for 2 decades, only to realize that maybe it's not all about volume, and it's not, bigger is not always better in terms of, getting an outcome, and finally working out some of the immunobiology related. And if we can identify neoantigens in a morphing tumor that are targeted and treated in a specific and ideal fashion that only focused ultrasound can do, and uses some feedback loop that MR can see, perhaps, now you're talking about a real paradigm shifting technology that has incredible impact. And I think we got to learn, it's not all about one aspect of getting an improved technology or improved technique.
Okay, before we are out of time, but I want to take 1 minute from each one of you to maybe tell us, summarize what you think your vision is for the technology. So I start with Kullervo. Go backwards.
Go faster and go better, do better. So.
Great. Well, very well said. Graham?
I think, as I mentioned, I think having more clinical trials with a standardized readout and maybe more standardized readouts, not just MRI-based readouts, would be a good way to go.
Great, thank you. Brad?
Don't become a technology looking for a home and find out a way to leverage drug plus device plus image in order to mechanistically immunomodulate or otherwise improve delivery of agents that are already known to work in a way that's unique and has impact, and have everything have a clinical impact and a road to clinical impact, specifically driving every decision we make along the way.
Gary?
Yeah, and continue to think about Star Trek. I still like that vision, right? And think about how focused ultrasound fits into the ultimate Star Trek Enterprise sickbay, right? Adjacent to the other technologies, because I can imagine focused ultrasound being one tool set, right? Next to immunotherapy, to Brad's point, to RFA, to other technologies. So how does focused ultrasound fit in the bigger puzzle? And then continue to focus on the focused few, no pun intended, on the targets we're doing well on, like double down on those, essential tremor, et cetera, et cetera. Continue to gain trust in the technology.
And ultrasound is one technology you can take to space. MRI, I don't know, maybe one day.
You know, that's a good point there. We should call Elon Musk and team to
Right.
make sure he knows about it. I think that's Neil's job.
That's right. Okay, Andreas?
Yeah, I think we also should take into consideration there is one MRI for tons of diagnostic procedures. There are ultrasound machines for same. Why do we have all these dedicated, very specialized focused ultrasound machines? So we should look for a multi-platform, multi-modality, and multi-use ultrasound, focused ultrasound system. And technically it's possible, and then it can be approved for certain indications through this, and makes it also through this process easier to handle. It's very complex, so those of you who know procedures, takes a lot of training, it's not standardized, and this is something one could rather solve with a multi-accessible focused ultrasound system.
Peiji?
So I think I've got Gary and Andrea streaming with me, so I'm glad to hear that. My vote is that, Clara, when you've built this system that I just described, let me know. We'll buy one at Stanford. We want to use it for sarcomas, pancreas, some ovarian cancers.
As you speak.
I'm being serious. I think it's not so crazy, and I look forward to testing your first version.
And yeah?
I'm not such a dreamer, so my takeaway is that I'll go for accelerating the treatment and go to fast treatments once I know what treatment is needed, and I don't think that we are at this stage yet. So.
Well, on this note, I want to thank all the panelists and the audience, both virtually and in this room. And I think the next stop is building the enterprise, right? Okay. Thank you, guys. Thanks.